Imagine being able to see the intricate details of a tiny cell, the building block of life. Thanks to microscopy, this is possible. Microscopy is a fundamental technique in biology that allows scientists to study cells and their structures in incredible detail. It all began with the invention of the microscope, a pivotal moment that transformed our understanding of life at the cellular level. Microscopes enable us to observe objects too small for the naked eye, such as cells and microorganisms. There are various types of microscopes, each with unique advantages. Light microscopes use visible light and lenses to magnify specimens up to 1,500 times, ideal for viewing live stained cells. However, their resolution is limited by light's wavelength. Electron microscopes, on the other hand, use electron beams. Transmission electron microscopes, TEM, offer higher magnifications, revealing cell organelles, while scanning electron microscopes, SEM, provide detailed 3D images of surfaces. Preparing specimens involves fixation, sectioning, and staining, enhancing visibility. Magnification enlarges the specimen, while resolution is the ability to distinguish 2 points separately. This microscopic revolution has been crucial in scientific discoveries, such as the cell theory.